Today we are going to talk about plant cells. So what are cells? The term cell originated from the Latin word cella, meaning storeroom or chamber. They are the basic building blocks of life, or they are the smallest living unit of an organism. Cells are capable of independent existence and propagation. Thus, it can grow, reproduce, use energy, or respond to their environment. Also, a cell may be an entire organism, such as the bacteria and the protist, or it may be just one out of the billions of cells that makes up the organism, such as the plants. The focus of our lecture today is the plant cell, and the major component of a plant cell is the cell wall. So cell wall is the rigid but elastic outermost covering of a plant cell. It provides strength, form or the structural support, and protection against infection, mechanical stress, and osmotic stress. It also allows cells to develop turgor pressure. So, turgor pressure is also called the hydrostatic pressure. This is the pressure of the cell contents against the cell wall, or the force within the cell that pushes the plasma membrane against the cell wall. So, cell wall is made up of several components, including cellulose, hemicellulose, lignin, pectin, soluble proteins, enzymes, and water. Cellulose is the primary composition of the cell wall. It is responsible for the elastic property and for maintaining the cell structure. Cellulose are tough, fibrous, and water-insoluble polysaccharides. So, hemicellulose is the second most abundant polysaccharide next to cellulose. So, it contains amorphous and highly branched short sugar chains that strengthens the cell wall together with cellulose and lignin. Lignin is a natural aromatic or phenolic substance that enhances the plant cell wall's rigidity and hydrophobic properties. It also promotes mineral transport through vascular bundles. Also, it serves as a barrier against pests and pathogens. So normally the phenolic substance are a group of metabolites derived from secondary pathways in plants that is part of the protection system. Pectin is composed of polysaccharides that aids in cell addition and hydration. There are three layers of the cell wall: the primary, middle lamella, and the secondary cell wall. The primary cell wall is the flexible layer. So it is thin and not rigid. It stretches as the cell grows. The middle lamella is where the pectin cements adjacent cells together, which prevent the cell from migrating. The secondary cell wall is the rigid layer. So this is thick and they owe their strength and toughness to lignin. So cell membrane, or also called the plasma membrane or plasmalemma, it has selective permeability. So this is the membrane just after the cell wall. So this part. So since it has selective permeability, it allows the cell to take up and retain certain substances while excluding others. So what are the components of the cell membrane? You have the phospholipids, proteins, and carbohydrates. The phospholipid component are arranged in two layers. It's what we call the phospholipid bilayer. So it has a hydrophobic tail and a hydrophilic region. Cytoplasm is the entire content within the cell membrane. So all of this is the cytoplasm. The cytosol, it is the gel-like or the fluid wherein all the organelles are suspended. So this is the cytosols. And these are the organelles, and we will discuss that later on. So the function of cytoplasm is for distribution of substances absorbed from the outside and distribution of substances released by the nucleus and other organelles through cytoplasmic streaming or cyclosis. The cytoskeleton is a network of polymers in the cytoplasm consisting the microtubules and microfilaments. So this structure helps maintain the shape and internal organization of the cells, and it also provides mechanical support that enables cells to carry out essential functions like division and movement. So what are microtubules? These are long, thin, and cylindrical structures. Each microtubule is made up of a subunit called tubulin. So microfilaments, these are composed of two actin chain, and it plays an important role in cytoplasmic streaming or cyclosis. So when we say cyclosis, it is the flowing movement of cytoplasm. Endoplasmic reticulum is the flattened disc or tubular sac. So these are the endoplasmic reticulum. There are two types, the rough and the smooth endoplasmic reticulum. So the rough endoplasmic reticulum are those with numerous ribosomes, while the smooth endoplasmic reticulum lacks ribosomes. The smooth endoplasmic reticulum or the ER is for lipid synthesis, while the rough ER is involved in the synthesis of membrane proteins and secretory proteins. In general, the endoplasmic reticulum is involved in cell-to-cell communication via the plasmodesmata. And in specialized cells, it serves as a storage site for protein. Golgi Apparatus is the collective term for all the dictyosomes. So dictyosomes are net-like structures of flat hollow disc called cisternae. So dictyosomes are also called the Golgi bodies. These are involved in the synthesis of polysaccharides and glucose units for cell wall formation, and they also secrete protein and carbohydrates from cell to the exterior. The mitochondria is a small and cylindrical organelles enclosed by two unit membranes, so the outer membrane and the inner membrane. The outer membrane is permeable for smaller particles, and it contains the respiratory chain component and enzymes for ATP synthesis. The inner membrane is impermeable and folded into numerous cristae. So the major role of the mitochondria is the synthesis of ATP through the process of respiration. Plastids are double membrane bound organelles involved in synthesis and storage of food. So there are several types of plastids: the proplastids, ichthyoplasts, chromoplasts, chloroplasts, leucoplasts, and many more. The first type are the proplastids. So these are undifferentiated, colorless, and tiny in size. And they may be differentiated into ichthyoplasts, chromoplasts, chloroplasts, and leucoplasts. So ichthyoplasts, these are specialized intermediate plastid types that are mostly found in dark-grown seedlings, and they may be converted into chromoplasts and chloroplasts upon light exposure. Chloroplasts, on the other hand, are the green plastids, so their color is due to the pigment chlorophyll. These are the plastids found in photosynthetic organisms. So chloroplasts, they contain multiple stacks of disc-like single lipid layers called thylakoids, which form a grana or the granum. So chloroplast is the site of photosynthesis. Since they have the ability to turn light into energy, they are also involved in amino acid and fatty acid synthesis, and they provide space for temporary storage of starch. Chromoplast. Chromo from the Greek word meaning color. These plastids are responsible for the yellow, orange, or red color of many flowers and other parts of the plants. So the functions of the chromoplast includes attracting pollinators, and it also serves as a storage of carotenoids and hydrophobic metabolites. Leucoplast, so leuco meaning white. So these are non-pigmented or the white plastids with storage function. So there are different types of leucoplast according to the substance they store. The amyloplast is for starch since amylo means starch. Elayo means olive oil, so these are for fats and oils. Proteinoplast is for protein storage. Another term for this is the aleuroplast and proteoplast. Another type of plastid is the gerontoplast. So these are chloroplast-derived plastids adjusted for recycling of plastids which are mainly found in senescence process or under stress condition. There are also several special types of plastids. The desicoplasts, so these are plastids that can be interconverted between chloroplasts and proplastids, and this is found in desiccation-tolerant plants. The phenyloplasts, these are phenol-enriched colorful plastids. Cytoplasts are specialized plastids in secondary vascular tissues that are dedicated to the synthesis of the precursors of monolignol production. The ribosomes, these are small particles about 17 to 23 nanometers in diameter containing unequal polyribosomes or polysomes. So polysomes are strings of ribosomes attached to the messenger RNA. So the function of ribosome is for protein synthesis. Vacuoles are membrane-bound organelles enclosed by a unit membrane called tonoplast. It is also called the dumping house of the cell. The functions of vacuoles includes absorption of toxic products, storage of various metabolites, and maintain water balance. It is also involved in the breakdown of macromolecules and in the recycling of their components within the cell. So, in mature plants, vacuoles are large and are extremely important in providing structural support, as well as serving functions such as storage, waste disposal, protection, and growth. Also, in plant cells, vacuoles are used to adjust their size and turgor pressure. Nucleus. So this is a membrane-bound organelle that contains the genetic information. Aside from storing the genetic information, it also controls the activities of the cell by determining which protein molecules are produced and when they are produced. So nucleolus is an area inside the nucleus that is made up of RNA and proteins and is where ribosomes are made. The nuclear envelope, on the other hand, is a highly regulated membrane barrier that separates the nucleus from the cytoplasm.